Good day, Jitney Techies, and welcome to this video where we're going to be looking at an exam question 3. So this is an additional question that we're going to be working through for our section on meiosis, just to help you prepare that much better for your final exams at the end of the day. So thanks for joining me. I'm Mr. P. I'm going to take you through this question over here. So you can see question 3 starts with an image and then gives us a little bit of context. It says the diagram below represents a phase in meiosis. Doesn't say what phase in meiosis, but it does, however, say that this is meiosis. Further to that, we've got cell 1 undergoing division that gives rise to cells 2 and 3. And then on the chromosomes, you can see that there are some alleles that are indicated by letters. So let's have a quick look at this. So if we look at the first 2 chromosomes, they carry the same allele on each. And at the bottom, we've got a homozygous dominant T with a homozygous recessive T on the other chromosome, which is going to mean that when this cell divides, we're going to have the movement of these chromosomes to opposite poles, and then they're going to be present in either cell 2 or 3. Okay. So that's important to remember. So we've got some context. So we're looking at meiosis, we're looking at cell 1, and we're looking at these alleles that are going to be separated during the process. Whole chromosomes are moving to the poles. That means what phase of meiosis are we looking at? We're looking at meiosis 1, and what would be when these chromosomes start moving would be anaphase 1. Okay, so that's an important one for us. So let's look at the questions quickly here. Explain why cell 1 is not or does not belong to a human being. Well, we saw from the image there that cell 1 only has 4 chromosomes in total, or 2 pairs, and humans have 46 or 23 pairs. So that would definitely make cell 1 a cell from a different organism than a human. If we look at 3.2, how many chromosomes would be present in? Here we're talking about cell 2 at the end of telophase 1. So we're looking at the end of meiosis 1. And if we recall from the process, we see that this is the first part of reduction division. So it's at this point now that I go from 2n to n number of chromosomes. So I had 4 chromosomes. I should have 2 chromosomes by the end of telophase 1. Next question, B. The daughter cells produced in cell 3 after meiosis 2. Now, after meiosis 2 means I've got the formation of the gametes already. And if you can recall that reduction division takes place after meiosis 1, where we have n number of chromosomes, and then it remains n number of chromosomes all the way through meiosis 2 until we have the chromosomes uniquely coming together in each of the gametes. So again, here my answer is 2 chromosomes. Now I like this question 3.3 because we get given a very, very unique image. We have to use this image and draw a cell diagram, or a label diagram, my apologies, of a gamete, so 1, that will result from cell 2. Now if this is meiosis 1, it means cell 2 is going to undergo meiosis 2 again to result in 2 daughter cells. That's just for cell 2, because that's the cell now that we are looking at. And that means that I'm going to have just those chromosomes moving towards cell 2. So I can't refer to the other 2 that I have encircled there. I'm only going to be referring to those 2 chromosomes. Now, we see that they're moving into cell 2, and when we enter meiosis 2, they are going to be pulled apart at the centromere, and I'm going to have in each one of the resulting gametes of cell 2, 1 copy of each of those chromosomes that are now joined by the centromere currently. So if I had to draw this diagram over here, we start here with the cell membrane. Now we need to understand that I'm drawing a gamete, which means I do have a nucleus, but I'm not going to put it in yet. I'm first going to draw my chromosomes that result. So there is my 1, and I mustn't forget to indicate the allele that has been labeled for me. Right, now we draw the next one, and again we are going to draw it exactly as we see it in cell 1, so as not to give the marker or the examiner any reason not to give us these marks. We see here that it's supposed to be a labeled diagram as a result of 2, so let's carry on and let's label and give this diagram a heading. So there we've got a gamete of cell 2, and when we get to labeling this diagram, we can label the nuclear membrane, we can label unreplicated chromosome, and we can label centromere. We can label cell membrane as well, but we look to put in at least 3 labels in a diagram like this. Generally speaking, we could be asked for 3, we could be asked for 2, but it just asks us to label. So my suggestion is to give at least 3 labels that you know are correct in your diagram. Okay, Junior Tuckies, thanks for that. Thanks for joining me for this video. Did a little bit of an exam technique here, looking at the question, how to analyze the questions, and then taking it forward into answering these questions. You're going to see that as we move through these exam type questions, there are a lot of similarities between them because they expect certain skills from us. So it's very, very important to take these questions, to go and practice them, and then to make sure that you're prepared for the questions that you're going to get in your exam. All right, thanks for joining again, and I do hope to see you in future videos.